Every year, over 1 million people die from cancers originating from mutations of the RAS gene. This process begins with the epidermal growth factor, or EGF, which initiates the growth phase of the cell cycle. It binds to the EGF receptor on the cell membrane. Downstream of this signaling pathway is the RAS protein. In a normal cell, the RAS gene codes for a small monomeric G protein that is a type of proto-oncogene. The RAS protein activates signaling pathways that regulate the cell cycle, mostly by inducing cell proliferation and differentiation. This process can be represented by a negative feedback cycle. First, RAS binds to GDP in its inactive state. The protein GEF catalyzes the release of GDP, allowing GTP to bind to RAS. RAS becomes activated to induce cell proliferation signaling pathways. Then, RAS proteins, with the help from GAP proteins, hydrolyze GTP to make GDP, returning RAS to its inactive state and preventing further cell proliferation.
But how do mutations in RAS lead to cancer? Genetic mutations in the RAS protein cause it to bind only with GTP, meaning that it is constantly activated. This creates a positive feedback cycle as cell growth signals are continuously sent, leading to the development of cancerous tumors. Screening for cancer should begin at around 50 years, with methods including imaging scans, blood tests, and biopsies, especially for anyone who has symptoms. This is so important because the deadly RAS mutation is present in 22% of all human cancers, specifically lung, colorectal, and pancreatic cancers. Meanwhile, scientists are developing solutions to flip the switch stuck in the on position.